Good evening, everybody, and welcome to our first UCD in Conversation webinar series. My name is Nicole Black and I'm the Director of Alumni Development here at UCD. It's wonderful to have so many of you from the UCD community here this evening to join us for the very first webinar that we've had, and I've no doubt that it's going to be an absolutely wonderful conversation. At UCD, building a community for nearly 300,000 global alumni and friends has always been at the centre of the work that we do. And we have traditionally done this, as many of you know, by welcoming you to events either on UCD campus or globally. However, it has always been the goal of the alumni team to diversify the way we engage with our global alumni community and friends. And the current crisis and restrictions on public events has given us the opportunity to do that. So now, thanks to the world of digital, our alumni community is more connected than ever before, and we have the opportunity to engage online with you in ways that we haven't previously. So every Thursday, I should say, through these webinars, we will welcome UCD alumni and academics to share stories of the tremendous work and impact that they have on society and to highlight the efforts that UCD is making in the fight against COVID-19. The webinar series is going to reflect UCD's new strategy entitled Rising to the Future, and its 4 strategic themes which focus on UCD's contribution to global challenges will carry through the conversation topics every week. Creating a sustainable global society, transforming through digital technology, building a healthy world, and empowering humanity. Before we start tonight, though, I would also like to take a moment to thank the alumni community and friends who have mobilised to support UCD at this difficult time. So as many of you know, UCD has been at the forefront of efforts in fighting COVID-19 in Ireland. And as a result of this, in the last week, UCD Foundation has launched an emergency appeal in support of the ongoing response to COVID-19 in an effort to raise €1 million to help save lives and support UCD students through the UCD Student Hardship Fund. Many of our students have never faced financial challenges like they are currently. So more than ever, we do need the support of the alumni community. And if this is something you'd like to learn a little bit more about, please go to www.ucdfoundation.ie tonight. So now, the format for this evening's event, it's going to include a 30 to 40 minute conversation, followed by a 10-minute Q&A session. And we expect to wrap up around 8:15 or 8:30 this evening. Please do feel free to submit through using the Q&A function at the bottom of your screen any questions that you have. But due to time constraints, please understand that Professor Gallagher will only be able to choose a selection of the submissions. If for whatever reason you have to drop off the conversation tonight, or you have a problem with connectivity, which I think many of us have had this week, we will be recording this session, so you will have an opportunity to view it again at a later stage on the UCD Alumni YouTube channel. So now, without further ado, I would like to hand over to Professor William Gallagher, Director of the UCD Conway Institute of Biomolecular and Biomedical Research, who will join us in conversation with UCD alumnus David Cavanagh, Director of Clinical Partnerships at Genomics Medicine Ireland, to discuss our genes and the battle with chronic disease and COVID-19.
Hello, everybody. My name is Liam Gallagher. I'm the Director of the Conway Institute. And thanks very much for participating in this event this evening. I'm just going to briefly introduce our guest today, who's someone I know pretty well, Dr. David Cavanagh. And David is actually currently the lead on a clinical partnerships team within Genomics Medicine Ireland, but actually carried out his PhD within University College Dublin within biochemistry, working in the group of Professor Therese Kinsella, but also spent some time in Systems Biology Ireland, and then went on to further afield. So again, thanks very much, David, for agreeing to speak on this inaugural event for UCD in Conversation.
No problem, Liam. Yeah, it's a pleasure to be here and delighted to participate. Thank you very much for the invite.
So we're kind of living in strange times. I see the background there, so I maybe give me a bit of a context there. I'm trying to predict what it is in Ireland.
Yeah, well, some people may recognise it. This is I'm broadcasting from the top of Torque Mountain in Killarney. Myself and my wife, we live down here, so this is one of the. I've been treating my colleagues each week with a new scene from Kerry. So we're we're blessed to have such wonderful scenes here. So this is the view of the Lower Lakes in Killarney for those of you that don't recognise it.
So we'll get into kind of the main topic a bit later on, but I suppose if you want to just fill me in a bit about, I suppose, your time that you spent in UCD and your experience there. Do you want to kind of go through that and give me some?
Sure. Yeah. I mean, I have wonderful memories really in UCD, and you know, quite a long association really with the college. As you said, I did my training with Professor Trace Kinsella, really focusing on cardiovascular research. And previously actually an alumnus at degree level undergraduate from University College Cork. So my home is actually, should we share the same home place in County Waterford, proud Waterford man. Did my degree.
I always thought you were from Cork. I
know. I'm right on the border. So I know I'm a mix of Kerry, Cork, and Waterford. So
the key thing is, what hurling team do you support?
Well, yeah, it depends on who's doing well in a given year. I'm lucky enough to have a choice each year. Yeah. So I spent a number of years do my research there with Trace's group. And then actually, towards the tail end of that, I had an interest in a kind of a startup idea that I pursued for a number of years, which was a technology startup company looking at. It was kind of an interesting mix of search engine technology, and we also trying to leverage the scholarly record or scientific literature to enhance scientific search, particularly as a focused on research reagents. So really what we were interested in developing was a way for scientists to find reagents to use that they could rely on, and also identify methods from the method sections of research papers. So we use a lot of, this is going back several years, we incorporate a lot of what's called natural language processing technology to index the entire corpus or a very significant proportion of scientific literature to enhance the record of scientific material. So as you look for, say, an antibody, you could see the full record of that antibody's use throughout the world, really. So spent several years developing that company, and then sold it to a company in the US called Compare Networks, who are based in South San Francisco, and spent a few years then kind of consulting for them. And from there, then
You didn't get the accent anyway, didn't you?
No, I didn't get the South San Francisco accent. No. And couldn't do a very good impression of it either. Yeah. Kind of throughout that period, then I guess I was looking for the next move for me. I've always kind of had a bias or an affinity, I suppose, for startups in general. I enjoy startups. I enjoy that kind of creative environment and working with, I suppose, often chaotic environments to start, but they are tremendous fun and tremendous kind of intellectual challenge. So I've always kind of gravitated towards those types of working environments, and I actually happily found such a place also in UCD with Professor Walter Colch and joined his kind of management team at Systems Biology Ireland, and spent several years then kind of working with Walter to, again, really, I was principally interested in the next thing. So trying to look at, I suppose if you look over my career, I have a recurring theme of this interface between technology, information technology and biology. I've always had a kind of an interest in that kind of nexus between those two things. And systems biology, of course, is very much at the nexus of informaticians working side by side, but with wet lab experimentalists, and so that was a great environment actually to cut your teeth in terms of kind of learning more about how biology was really moving from, I suppose, historically kind of a small groups doing very focused things for pretty much their entire career. Groups would pursue their pet proteins to much more of an orientation towards team science, big data sets being used to uncover and answer questions that maybe previously hadn't been possible. And before I did, so then I left UCD for a couple of years, and I spent probably 2 to 3 years working with a group in a medical device software company, again really to develop my was interested in developing the skill set around software and life sciences. And S3, particularly good at medical device software, you know, and I kind of learned a lot, worked on a big pharma. They had a pharma client based out of Switzerland. So I spent several years working as an analyst on a project there, which brought me to the States a lot. I actually spent a lot of time studying kind of healthcare environments in the States, particularly around MS care and rheumatoid arthritis, which was great fun, great learning experience, and really got to see how healthcare is delivered in the US, which is a really interesting experience. And then I guess I got to hear about GMI. I kind of looked at GMI, and I've been watching it, and of course I knew some of the founders. Very strong link to UCD. Obviously, Sean Ennis, as one of the founders, is a UCD colleague, and was kind of intrigued by the ambition of, I suppose, of the project. You know, I always had an interest in genetics and population genetics, and thought it was kind of cool that, you know, company was trying to take on a large project or an ambitious project like that. And the decision to join was a kind of a difficult one. But sorry, yeah, Liam.
No. I suppose I just want to kind of, tap into that, this idea of the convergence of, I suppose, computer science and biology.
Yeah.
And this, now the onset of, I suppose, artificial intelligence technology, I suppose to a certain degree, it's probably relabeling of things which were already probably being done, but maybe with the new power of computation.
I think that, yeah, go ahead.
And I'm just wondering, in terms of, it sounds like in your career that you've kind of been drawn into those kind of areas, I guess, in terms of that intersection.
Yeah, well, it's funny. I mean, I have, and I've always been intrigued by it. I suppose I kind of grew, you know, I even undergrad, one of our colleagues, Des Higgins, actually taught, as I was taught. Des taught me as an undergrad, and he taught bioinformatics in UCD for several years. And I was just before the cusp of this. You know, it was pre the first human genome project. It was pre-human genome project. You had kind of first generation technology for studying the genome. So there hadn't been a whole genome done in my kind of formative years in terms of scientific training. But the writing was on the wall. You know, there was a sense that actually it was going to become very much a data science. You know, biology was following physics to a large extent, and interestingly, I thought it was really great last year. It was last year that we had the Schrödinger celebration. And of course, you know, Erwin Schrödinger wrote that fantastic book When He Was Here, What Is Life, and he was very much applying physical science thinking, the principles of physical sciences, to biology, which was 50 years ago. But the technology really has only matured sufficiently now to really see convergence between biology and big data science. And I think I feel privileged to be kind of lucky enough to be a scientist at a time when these fantastic things can be done with biology, and you know, you can apply the rigor of mathematics to understand biology in a more fundamental way. You know, I think it's great.
So look, if you look at, I suppose, UCD's new strategy, one of the kind of key areas is building a healthy world. And obviously, you know, one of the challenges we face, and we'll come on to COVID a bit later on, but obviously it's the area of chronic disease. For example, you know, I work in the cancer space, and we kind of interact at a certain level within this new program, Precision Oncology Ireland. I don't know, in terms of your ongoing work and the work that you're doing in GMI, do you want to just tell us a bit about that kind of focus on chronic disease? And
I think one of the challenges is there's been a lot of false dawns in precision medicine, and it's kind of one of those things that I guess is, the term precision medicine has been used for a long time, and it's almost like it's always just around the corner. But I think, you know, certainly in the last several years, you know, since the Human Genome Project, you're starting to see application take form in more tangible ways. And certainly, you know, the space that you're in, oncology, genetics has really played a bit of a transformative role, not just in terms of diagnostics, but also in terms of, you know, targeted therapies and so on. And it really has been a kind of the first therapeutic area, I'd say, that has been a benefit of this really, really targeted precision application of medicines, or identification of populations that would benefit from treatments. Similarly, the other area, of course, is rare disease, pediatric disease, where there's immediate, and the reason for that, of course, is there's very discrete mutations that are existing. There's not lots of mutations, very well what are called highly penetrant mutations that have high impact and will show up with small numbers, right? When you study it. The challenge with complex diseases, and we have studies examining a range of complex diseases. For example, one of our longest standing studies in Ireland is an MS study. The challenge with MS research in general is it isn't one gene or two genes that are increasing your risk. In fact, it's not even thought of as a genetic disease in many respects. It's more kind of always been regarded as an inflammatory disease, and actually, but there's been excellent foundational work done by the Wellcome Trust Case Control Consortium on MS. But what that has shown, in the last 10-15 years is that, it's not 1-2 genes, it's 200 or more genes that are having small effect sizes. So small contributions that are tuning the body's response or tuning your risk of getting MS. And also, of course, what's really even less understood now, so I think some of the great work that's happened in MS genetic research in the last several years has probably established quite well what your lifetime risk of getting the disease is, but very little is known about, if you have MS, why do you progress and have some people have good outcomes, other people have bad outcomes, and this is where population genomics can play a really significant role in bringing us to the next level, I think.
Apologies, there's a bit of a dog, probably hear a dog in the background. Apologies for that. So this is one of the benefits of doing this from home. So one of the things I want to ask is really kind of, I suppose that's interesting. You've got, obviously, you can have this background genetic information. How will you use this information going forward? Like, how will this be mainstreamed into clinical practice, or can you use this information to develop novel therapies or diagnostics? What's really the purpose behind this, I guess?
Yeah. Well, I mean, the good news is it's already been used. And I think, again, just drawing on the examples of oncology, for example, it's well known that there are treatments that are only effective in people that have certain mutations. So there are certain types of breast cancer that will only respond, people will only respond to drugs who have where those mutations are present. So, and of course, in medicine, one of the guiding principles is do no harm, and the knowledge of medicines being ineffective or in some cases harmful if they're taken, it's obviously not a good thing to prescribe a medicine that is not going to work. So I think that's where you're already seeing the application of genetics, this fundamentally this understanding that we are all individuals, and what we regard as the same disease often isn't the same disease, and genetics is making it easier to separate out populations that share common genetic factors that either drive their disease or cause them to respond to drugs in different ways. And I think that's already having a benefit in terms of patient outcomes. All medicines fundamentally are not something that the body wants to take, or we take medicines to make us better, but they do have side effects. And if you don't have to take a medicine, or if you don't want to take a medicine that won't work, I think genetics gives you the tools to allow that to be untangled more fundamentally.
So again, if you look at the kind of current crisis, I suppose we're in internationally, COVID-19, obviously that really has really shown that life science diagnostics plays a huge and an important role in that context. And I just want to know, I suppose, in terms of, your own kind of changing work practice, or the kind of work that you're involved with, has it been affected by COVID-19 and for example, the work that GMI might be doing in that space?
It has, I mean, in very fundamental ways. I mean, I suppose, obviously, as we're here together tonight, all our staff are working from home, both in Ireland, all our staff work from home with the exception of our lab staff. But globally, our organization, everybody in Boston and Iceland, are all remote working. Thankfully, we've adapted, I would say, pretty well. We're having fun with Zoom and Zoom backgrounds, and I think there are benefits to working from home. I think people There are challenges with it, no doubt, but I think there are also many benefits. So I think fundamentally it's probably going to shift how we work, how we all work post-crisis. Day to day, then, I think in terms of our business, we've certainly been impacted. Much of our research is obviously dependent on our collaborations and being able to partner with hospitals to build cohorts collaboratively. And really, research globally, clinical trials or the type of research that we do, which is observational research, has been stopped, right? So they don't want people in the hospitals that don't need to be in hospitals. So, I think that's going to create some problems. I think, for example, clinical trials, I think you're going to see a delay in drugs coming to market as a result of this crisis. I think it's one of the kind of collateral damages. I think this crisis will mean that drugs that might have come to the market in a year's time will now not come on the market for 2 years, for example. So that has certainly presented some challenges, I think, for us as a business.
So obviously we've seen within an Irish context, but also internationally, people have really focused specifically on ramping up testing for the virus.
Mm.
And obviously within Ireland, we've obviously set a quite ambitious target of something like 15,000 tests per day by, I think, the middle of next month. And we're pretty much on course for that, but that really has involved a huge degree of partnership between academic partners, industry within Ireland, and obviously the clinical labs. And I think GMI, for example, have played a key role in that in terms of helping, say, the ongoing testing activity. I don't know if you wanted to talk about that.
Yeah, I mean, we when this crisis hit, I guess we were, I suppose, similar to everybody in Ireland. I think it was really inspiring to see the level of community and really the goodwill, the outpouring of goodwill, I would say, from all sectors of Irish community in terms of making a contribution and wanting to do something. And of course, we were no different. Many of our staff in Dublin feel very passionate about being able to help Irish people and help the system in a time of crisis. So we were trying to kind of figure out how could we make a contribution. I suppose some of the areas of competency that we have, in our facility and with our team. And I would have to pay tribute really to Patrick Buckley and his leadership here in regard to the support we provide for testing. But for example, our laboratory is highly automated. So we have a lot of technical expertise in terms of scale up of assays and automation of assays. So I think that was, Patrick has a kind of long-standing kind of collegiate relationship with some of the team at the NVRL. And obviously we have a strategic partnership with UCD, which we're very proud of. So that was an obvious kind of piece of knowledge or expertise that we were really happy to share and participate in. And then it became apparent, of course, that, I think one of the things that happened was, it was very easy to get people sampled for testing, but there was a global shortage of the components that allow you to process samples and run the tests. So what we were able to do, I guess, is, the easiest way for non-scientists to describe it is, we were able to source some of the ingredients for the cake and make some of the components that could be used to scale up an assay in partnership with the NVRL and some other private sector companies. And so really our contribution is reagents. So we're not involved in the testing per se, but we are providing reagents that allow scaled up testing, which hopefully will satisfy the need for, the contact tracing efforts.
Yeah, actually, it's quite interesting because I was participating in a webinar earlier on today where Cillian de Gascon, the director of the NVRL, was speaking, and he was saying that I think the PCR reagents to detect the virus, initial paper on that came out on something like the 21st of January. It was submitted, was accepted the next day, and then printed the next day, and the information was then sent to NVRL. And I think within 2 weeks they had an assay designed.
Yeah,
it was amazing the rapidity or how quick that information has been transitioned into a clinical assay. Yeah, and
it's really incredible. Yeah.
One thing I wanted to talk about a bit more detail was, I suppose, the benefit of sequencing. And so again, the NVRL within UCD, actually in collaboration with ourselves at the Conway Institute, they actually sequenced some of the first patient samples that were in Ireland in terms of COVID-19 patients. And they actually found, interestingly, that I think one of the first patients, the sequence was 100% identical to the strain from Wuhan City, but the others were actually more closely related to the variants which are emerging from northern Italy. So this kind of sequence-based information gives you an idea of spread, where the disease is coming from. And I know that I think you're involved in a project which is Paul Cotter in Chagas has recently got funding for. So I don't know if you want to talk about that and maybe other aspects of sequencing in host genetics.
Yeah, so I mean, I guess there was kind of two main pillars to our response, I suppose, to the COVID crisis. One, obviously, is the testing support for the testing effort. And the other kind of strand of support, again, just looking at what we can do and what we can offer is we obviously have a lot of capacity to do sequencing. And if you look globally at the response to the COVID response in terms of sequencing, there's really two main things being done, right? One is sequence the humans, and the other is sequence the virus, and both are important and interesting for different reasons. So Paul Cotter's and that consortia is very interesting. You're right. I mean, I think one of the really important things to understand is COVID is a virus. It has nucleic acids, it's an RNA-based virus, and viruses mutate. Like all our genomes mutate over time, and the viral genome will mutate as well, and will give rise to strains that may have variation in terms of their how pathogenic they are, how infectious they are, how they behave when they infect people, and the responses that they can elicit in an infected human. So it's very important to be able to understand that evolution of the virus over time, and it's also very important to understand, whether the strain is getting potentially more virulent over time, or maybe its virulence is diminishing over time. So there are many efforts, and I think Ireland it's great to see Ireland take a lead in this and doing it I think one of the most encouraging things for me and the consortium that Paul has kind of, and Fiona Crispy has led, is that they've taken a national view. I think one thing I would say about using genomics and the power of genomics, scale matters. Doing things at scale matters. Small numbers will not help. Their studies will be underpowered. Conclusions will be made that aren't statistically relevant. So it's very important that people cooperate around to achieve scale. And we've some collaborators in the UK as well, where they're doing this really well. The Sanger is taking the lead. The Sanger is a major sequencing hub in Cambridge, and they have organised nationally to sequence the virus, and there's multiple centres collaborating around that. And it's great to see Ireland kind of coalesce around a consortium to do this. The other area then, of course, is the human. And we're involved in supporting a number of projects. Again, our involvement in many of these projects is we have technology and we're offering it to investigators and academics who are leading these studies. So we're very much an enabler of the research as opposed to taking any lead on a particular project. But I think what's interesting about the human sequencing is there is kind of evidence around the world that people are responding to virus infection in different ways, and that may not be down to the virus. That could be down to them. It could be down to some unique factors about them, and this is where genetics can help you understand. Okay, if you it's classic population genomics. Take a group of people who look, are similar age, similar gender, can be grouped together quite well. What is different? And let's say they end up respirated and severely ill. Similar group of people who have a similar infection but are not responding as severely. What is the difference between those two groups? And that's what genetics can help you kind of understand. But again, that's where really important to get the scale, to get your numbers high enough so that you can have confidence in the conclusions.
So I'm going to actually take some of the questions that we've had so far. And then again, I would encourage people to submit questions online. One of these, I suppose I can potentially contribute as well, maybe yourself. There's a question here from Jenny Blake. Do you think the criticisms of the low level of testing in the media is fair? I assume this is relating, I suppose, to potential criticism in Ireland. Again, I suppose I'll give you my perspective first and maybe get your opinion. is I think actually we're doing quite well. If you compare us per test per million population, we're comparable to Germany, which is suggested as being a high test rate country, but we're nowhere near somewhere like Iceland. Now, Iceland is obviously, it's a smaller country, but they obviously have a big track record in terms of indigenous industry within the molecular diagnostics. And obviously there's some kind of, connections that you have there. I'm just wondering what's your perspective of that?
Yeah, I mean, I think I agree. I mean, I think, we're in a kind of a once in a generation crisis. So, we have no precedent here to kind of benchmark ourselves off of, really. I think Ireland has responded really well, I have to say. I think we've organized quickly. Our communication strategy has been, the government's communication strategy has been really good. It's challenging to scale up from nothing to the level of testing that's required in any environment. I think what I would say is we shouldn't get complacent, or we shouldn't get self-congratulatory. I think there's a lot of work to be done, and it needs to be done quickly. So we should always be urgent in everything that we do. But I actually think, if we benchmark ourselves against, bigger and more, bigger countries that on the face of it have maybe better developed healthcare systems, I think we've done a pretty good job, I would say, and I think we should be proud of that effort. And you're right. I mean, I think Iceland, we have a connection to Iceland in that we share a co-founder of a company called GCODE, who really led the way in terms of population genomics. So our chief scientific officer of Mutations, of which GMI is now a wholly owned subsidiary, we're really at the forefront of this entire, kind of this field, and Iceland was, I guess, one of the years that population genomics became a mainstream thing, and GCODE still exists, and Carrie Stephenson still leads that company, and they've done some excellent work in the last couple of weeks and published some really interesting work in terms of on both the testing side and also some new genetic work as well. So.
I was actually reading some of his commentary actually, and he was Carrie Stevenson, and I'm not sure if he pronounces his second name correctly, but he seems an interesting character. But ultimately, there's some suggestion, at least when we're looking at children, because there's some debate about whether you should keep schools closed or not. But there's some suggestion that children really who are infected with the virus really find it very difficult to be able to pass it on to other individuals. And so maybe there's some scenario where maybe that model of closing down schools mightn't be overly appropriate. So it's interesting to see how that might pan out. I have a particular question here from a colleague of mine, Jeffrey Glennon, who's a new hire into UCD actually under the Ad Astra scheme. And it specifically probably correlates, does GMI essentially develop a resource similar to what GCODE genetics has done in Iceland? I suppose to a certain degree it's similar in terms of context, but I don't know, do you want to kind of comment on that question?
Yeah, I mean, I think we share some characteristics, I suppose, with GCODE. I mean, again, we're not GCODE, and Mutations is not GCODE, but we do obviously share a lineage, if you like, in that Jeff was a co-founder. Jeff Gulcher was a co-founder of GCODE, and GCODE had a focus on population genomics. I think probably the way to think about it is more in terms of the science. So one of the reasons that Iceland was a very interesting place to study population genetics is because the population there has been historically quite static. And what I mean by that is, if you go back 40 generations or so, Iceland actually was not an inhabited island. It was a vacant island, and it was inhabited actually coincidentally by some Vikings, some Scandinavians, who swung by in Scotland and Ireland. And if you look at the genetics of Iceland, the maternal line, and you can discover the maternal line through mitochondrial DNA, is predominantly Irish actually. So we have a historical link to Iceland that goes beyond just the Vikings visiting Ireland. Is actually a maternal line.
Visiting is a euphemistic term.
It's a euphemism, yeah, probably most likely. I doubt they went willingly, but we'll never know, I guess. nevertheless, I think what was the characteristics of Iceland that made it an interesting and exciting place to do genetics is that population structure was very static. They had a very strong ancestry and ancestral record, which allows you to kind of make quick. It kind of offsets this need for numbers, so you can make quick discoveries with lower numbers. And Ireland shares some of those characteristics. So we have, probably not to the same extent, it's in relative terms, we've not been as homogenous. We've had a lot more migration. And of course, you know, if you go back long enough, we're not even really an island, right? We were linked to the continent. So it's not, you know, to overstate it, but certainly from a scientific perspective, Ireland remains an interesting place to do this type of work because we are an island and we've had what are known as population bottlenecks as well. We've had the famine, for example, which basically shrinks your gene pool and makes your gene structure quite static, and again confers some advantage. But I would say as a company that we've evolved now as WuXi Nexco, we would see ourselves as population genomics enabler, really. So we don't just work in single populations. We don't just operate in Ireland. We are interested in collaborating with health systems and academic groups globally to apply population genomics to answer fundamental questions about biology. And that's not unique to just Ireland, but Ireland remains a very interesting place for us to do this work, for sure.
That's quite an interesting question from Niamh O'Brien. Regarding your work on mutations in MS, can CRISPR technology be used to address these, or are there too many mutations present for this type of technology? But obviously, I suppose more fundamentally, can CRISPR be used fundamentally for humans? But what's your ideas around that area of?
I mean, I'm not an expert in CRISPR by any sense. I mean, there's been a lot of excitement about CRISPR and gene editing in general. I think there's limitations to the technology. I think as it relates to MS, I think where gene editing, let's put aside CRISPR, which is a technology to do gene editing. So I think you have to think of those two things separately. But where you can effectively correct causal mutations in disease, that is already happening, right? There are diseases where causal mutations can be corrected using a variety of different techniques. Such as in hemophilia, for example, where it is actually making a difference. Yeah. Now, that is again going back to my earlier point about diseases that have small number of genes with large effect sizes. MS has hundreds of genes. Yeah. I think it seems, for me at this point and where we are, the notion of being, again, it's a little bit like fine-tuning your body. They're all small effects. So the application of gene editing in an MS context seems a little bit more challenging from where I stand, but of course, you never know. It's not our area, so it's difficult for me to speak from a position of authority in that front.
Yeah, so this is a bit outside my territory anyway, and it might be yours, but it's an interesting question from Gerald Barry. If sequence mutations are found in the virus, how will you know if these mutations increase or decrease the virulence?
That's a good question, yeah.
So I suppose, as you say, numbers, I suppose you will see some associations between certain variants and virulence, but...
Yeah, well, it's very, I mean, this is, again, it brings in, it kind of tees up a pretty interesting point about genetics in general as well. So one of the key, if you're studying any disease, genetics on its own, and a lot of genetics has been performed, really has looked at just the genome, right? But actually to make breakthrough discovery, you have to understand phenotype, and phenotype are the features of, you know, either cellular behavior or disease behavior. And I suppose as you talk about a virus, how does a virus behave in the body, and how can you actually study that, document it, and associate it with a mutation? And it's a really good question, and it's where phenotype will become incredibly important. Understanding the phenotype of the virus and that particular strain of the virus really becomes super important in understanding how the virulence has been affected one way or the other.
Yeah, so it's a question from Finton James. Is GMI's work mainly with hospital patients or with healthy people, or those with less severe chronic disease? I think it's a combination of those things, isn't it?
Yeah, I mean, we have a variety of different programs that we run either in the hospitals, or we have actually we have a study that we run in UCD called GeneFit as well. So our studies are all what I would say is the study design and protocols are all kind of independently reviewed by ethics committees. So, for example, our study in the GeneFit study in UCD has been approved by the UCD ethics committee to for the study to be run and for it to be offered to participants who are interested in volunteering. And that study is really kind of, you can self-refer into that study. It's a fitness study, so you can have a DEXA scan. And you volunteer to participate, and you volunteer for your DNA to be used to fundamentally study, assist in studying the disease, the genomic basis of many diseases, as it's used as what we call a population control. And then our studies in the hospitals, again, will go through a similar process of ethics approval with the local hospital ethics boards. And they are offered to patients as they attend their clinics. So, for example, in an MS clinic, the patient will be offered an information leaflet, outlining who GMI are, the fact that we're a commercial company, and so on. And they'd be invited to participate if they so choose. And they're the two main ways that we build our cohorts and we do our research.
So I'll have a question I'll come back to a bit later as we sum up, but it's a nice kind of summary question, but maybe think about it in the background. It says, what has been discovered so far thanks to the GMI initiative? It's a big question, so I'll let it percolate in the mind for a while. I have an interesting question from the famous Bim Tormley, I think. He has it's probably somewhat indirect question from our conversation, but it's I was trying to find it. Actually, it's gone off a bit here. He was talking about what happened to the 10,000 per day tests, which was kind of commented about. The government strategy is very poor. There's some comments around that. In a sense, I suppose I can kind of have some commentary on that because I'm involved in a group called the HSC COVID-19 Lab R&D Product Solutions Group. And actually, we are at a capacity, certainly at 10,000 a day. It's really, we're in that position. It's really ramping up to 15,000 per day. And again, I think that a lot of that is down to the I suppose, the good work that GMI and others have done in really providing materials for testing. Now, I'm just trying to think. Okay, one second. I'm going to put on my glasses if that's okay. This is the age-related issues here. Okay, I work in addiction medicine. Even though our population seems vulnerable with chronic health problems, none so far have contracted COVID-19. Okay, interesting. Any ideas? Not sure if we can answer this question. Any ideas why this would happen? Seems like inherent immunity. I doubt it very significantly, and one would, it's an interesting idea, but I first have heard of it. How can each country use their strength to help weaker countries on healthcare? It's a bit more broader question. I suppose to a certain degree, we've been in a situation in Ireland where we've actually, to a certain degree, have to help ourselves, because we've had this, particularly in the area of testing, to a certain degree, we're quite reliant on external parties for testing. And this is where we had to actually come together to actually resolve that. Certainly, we can actually transfer some of our knowledge that we've gained in that to other parties as they ramp up activities. I'm trying to think if there's any other questions here that might relate to our conversation. Okay, well, maybe you can go for that big question since it's pertinent in mind. And I suppose what's been discovered so far, and we might use that to kind of generally wrap up our conversation.
So, I mean, I think I think maybe the way to think about it, again, is we get asked this question a lot. What does the result look like for GMI? And it in some ways it's not terribly exciting, because we do a lot of work to get the numbers that are required to make good conclusions about what might be going on at a genetic level. So there's a lot of effort goes into to assembling very well powered data sets, and ultimately what you find at the end of it is if people are familiar with what's called a Manhattan plot. Manhattan plot is, it's a way of it's a way of representing the genome, and it's called a Manhattan plot because it looks like, the skyline of Manhattan, and it has 23 chromosomes. It has an x-axis of 23 chromosomes. And really what population genetics allows you to decipher from populations is what genes are more prevalent in your case group versus your control group. And that's it. That's what a result looks like. So it allows you to understand the differences between the two groups. So probably the most exciting areas for us, obviously, we're getting to the level of scale in MS where we're getting some interesting findings, I would say, but they're not conclusive yet. So they still require verification. They probably are still slightly underpowered to be at the level of confidence required. So that will tell us something about the genetic landscape of MS. I think that will be potentially it will add to the stock of knowledge about MS. I won't make a claim that it will be transformative in our understanding of MS, but it will help us to understand something about MS that we don't currently understand, which is incremental, I would say. The other area that probably is more for us is more short-term and tangible in terms of our results that you can point to is, of course, we do support rare disease program in some of the children's hospitals, and we've been really privileged to be involved in assisting some of the clinicians there in solving some pretty complex cases of kids who have not been lucky enough to get a diagnosis, where we've done a whole genome sequence, and some of our team will support the interpretation of those data sets to help make a diagnosis about what might be going on for this child. And that type of analysis is very satisfying. I can't always say that it leads to a treatment, but it certainly in a lot of cases, I think it's helped people understand that there is actually a disease here, right? So we've been able to kind of find a genetic basis for what their child is maybe suffering. So I think we've had some really exciting results there, and we've been thrilled to support the clinicians in solving some of those cases. So I think that's the kind of spectrum of results for us. Yeah.
So obviously myself, I'm a cancer biologist, and one of the programs I'm quite heavily involved with at the moment is this new program, Precision Oncology Ireland, which is funded by Science Foundation Ireland, but also 6 charities, but also GMI is a key participant there. And I suppose what we're trying to endeavour there is really have a team-based approach across Ireland to address key challenges in terms of, I suppose, precision medicine or introduction of precision medicine in the cancer context. And I suppose if you look at the application of sequencing types of technologies, as you commented earlier, you've really seen a fantastic impact in terms of on clinical care, in terms of a direct impact of sequencing technology.
Yeah, I think that's right, and I think cancer, and oncology in general has definitely been a beneficiary of the technology, and the advancement of the technology has been fantastic. We've been very excited. I think POI, and even a lot of this is credit to the work that you've done over many years, really, in terms of building. Supporting the development of oncology infrastructure and oncology research infrastructure, I think is in a 20, it's almost unrecognizable from what it was 20 years ago in terms of the cohesion and the levels of cooperation. And I think for me, working with Walter as well for many years, it's been a real privilege to be involved in POI. I think it represents a milestone in terms of the levels of kind of broad-based cooperation between industry, academia, and charity groups, and it's very encouraging actually to see that kind of coalition come together, and for people that have different sets of expertise to be able to, contribute in constructive ways to move the field forward and move Irish research forward, which we're very excited about. Okay,
look, I think I had a good chat, and I really like to thank everybody who has submitted questions. Apologies if I didn't get a chance to go through all of the questions. And again, thanks for everybody for participating. I'm going to hand, just before handing back to Nicole, I'd like to thank you, David, for for participating. And I'm a bit jealous because, again, even though I'm from Waterford, my favourite county in Ireland is so, particularly...
I'll send you some pictures, Liam, don't worry.
Yeah, Castle Gregory is a fantastic place. Yeah. So once I get through all of this, I'm going to be kind of skedaddling over there. Okay,
yeah, no problem.
I'll hand back over to Nicole to wrap up.
That's great. Thanks, guys, so much. That was an absolutely fascinating conversation. So, Professor Gallagher and David Cavanagh, I cannot thank you enough for launching the UCD in Conversation series and for sharing your time and expertise tonight. It's an area I personally know so little about, and I find it fascinating. And I hope that our audience at home found exactly the same as well. So as I mentioned just earlier this evening, this conversation will be available for future viewing on the UCD Alumni YouTube page. And if anybody tonight has any comments or conversation or comments or questions about this series, or any other just questions in general that you would like to ask the alumni team, please do email us at alumni@ucd.ie. So, as I mentioned, tonight was the first launch event of the series, and we have our second one next week, which is going to be next Thursday at 7 p.m. So we are very excited to welcome UCD alum Caroline Casey, an award-winning social entrepreneur and founder of the Valuable 500, and also Martina Fitzgerald, the political journalist and author who will be in conversation then. So thank you once again for being with us this evening, and thank you so much to William and David for such an interesting conversation. And we really look forward to welcoming you all again next week. And in the meantime, please stay safe. Thank you.
Thank you.